Nothing.
Thank you so much, Tom. I'm the director of the Center for Biomedical Image Computing and Analytics at UPenn, and I think it's great to follow up on both Paola and Andre. I'll be talking about some of our work on decomposing neuroanatomical heterogeneity via mainly semi-supervised machine learning, which is a way to actually partially deal with the uncertainty, with the difficulties of parsing heterogeneity in the sense that you have a control group, say the healthy individuals, who have some heterogeneity, but that doesn't translate into your ability to cluster disease effects. So you kind of factor that out if you want. And the second thing I would like to point out, and I think Andre made the point very strongly, maybe there are no clusters in the data. What I would like to emphasize is that all of this perhaps should be viewed as part of a dimensional neuroimaging approach, where instead of having umbrella diseases, we have axes or dimensions that indicate that you're more like this, that you more have this phenotype or that phenotype. The methods I'll be discussing have been applied also to functional data, to amyloid data, and so forth, but I'll focus more on entirely on structure. So machine learning is very popular, and it's been popular for a while, but particularly lately with the advent of deep learning. And some of the problems that Andre described actually have become a lot more persistent with deep learning, which can fit any data. This is some work we've been involved. We got involved a long time back in 2005. It was one of the first approaches where we found that there is an imaging signature of schizophrenia, and that was quite robust at the time. And since then, people have reproduced these findings in schizophrenia and in depression and other areas in terms of supervised machine learning and finding imaging signatures. And some of the signatures have been predictive of clinical outcome. However, there are many persistent problems, and I actually wrote an editorial in Neuroimage last year. I touched upon a few of them. These are two of them. One is reproducibility. Machine learning methods tend to overtrain in a particular data set, and they don't reproduce well in another one. And heterogeneity, which implies that if I go back, I showed you that there was an imaging signature of schizophrenia. Now we know, and I will show you some results, that there are two imaging signatures of schizophrenia. So it's a mistake to actually try to find the one signature if there are potentially multiple. So to address these problems, a couple of years ago, we put together this consortium, the Phenom Consortium, with funding from NIMH, and we're pulling together data from more than 10 sites all over the world, several from Europe, and Paula described some of them, Australia, Brazil, and Penn in the US. And we're trying to basically have a large enough N of data that we pull together, we harmonize, and that we can start, that can help us start addressing some of these issues of reproducibility and heterogeneity. So the first result was published about a year and a half ago here, where we were happy to see a reproducible imaging signature of schizophrenia across multiple sites. This was results from a little less than 1,000 people from 3 sites, Munich, China, and the US. And there was a fairly robust imaging signature that gave an AUC of 0.84 or so. And these are different regions of interest in which we found that there were diffuse cortical volumetric reductions, the blue, and increased volumes in a number of areas. Some of them are the ventricles, but 2 of them, and I would like to point your attention to that. One is the internal capsule and the other one is striatum. They were bigger in patients than controls. And this is something that we published here, and other groups like Enigma has published as well, and other studies. But I think we have all failed to recognize that these effects here, the increases, might come from a different patient subpopulation than these diffuse decreases that we consider to be the hallmark of brain structure in schizophrenia. And I'll show you now how with semi-supervised learning approaches, we actually learned that recently. So heterogeneity is a problem, and we've tried to address this problem by taking semi-supervised learning. Rather than being trapped by a lot of the pitfalls that Andre described in clustering, where we could be clustering according to sex, age, scanner type, interpolation method. It has happened to us. There was a method that was very well identifying a subgroup of people, and then we find out that they have been interpolated with a different interpolator images. So instead of doing that, we assume that there is a control group, which is denoted by Y here, and these are perhaps our healthy controls. And there's a lot of variability in these people due to confounding sources of variation that we don't want them to affect our ability to find clusters. And we built two methods. These are two very bright engineering students from Penn. One is Aoyan Dong, who is now in industry, and the other one is Erdem Varol, who is now at Columbia University. And both of these methods assume that there's a control group and there's a number of transformations in this approach that map this probability density function to a broader, more heterogeneous probability density function via a number of transformations that we don't know, but we're trying to find them out from the data using a generative approach. And the other one is similar in flavor, but is more discriminative. The control group is separated by the patients via a convex polytope that uses the Hint's loss function similar to what is used in support vector machines, and it uses an expectation maximization method to sort of simultaneously or iteratively estimate the clusters and the separating hyperplanes. Both have the flavor that they're trying to separate the control group from the more heterogeneous patient group via a number of transformations. And presumably these transformations now more clearly identify pathologic processes that moved this distribution to this distribution. So we first applied this, and I'm sorry, I'll deviate a little bit from the main theme of the talk, but we first applied it to people with mild cognitive impairment and Alzheimer's disease. We used data from the ADNI-1 and ADNI-2, 1.5 Tesla and 3 Tesla. We lumped together all the MCIs and the ADs into one category. These are the people who have cognitive decline, they have problems, and the normal controls were in our uncontrolled group. Now, ADNI is a fairly homogeneous group of individuals compared to what you see in the average aging memory clinic, because they've been selected to be very likely to have either Alzheimer's disease or amnestic MCI likely to progress to Alzheimer's disease. But even in this population, we actually found 4 subtypes, which we didn't quite expect. So 13% had completely normal structure. And that might be because they were people, most of them were MCI individuals. Most of them might be MCI for other reasons, or it could be that our control group, who are also older adults, of course, they might have had a lot of atrophy, but yet because of cognitive resilience, because of brain resilience, cognitive reserve, and other reasons, they didn't decline yet. But nonetheless, 13% of the people had completely normal structure. 44% of the individuals had, in the broader patient population, had what we expected. This is a typical Alzheimer's type of pattern of brain atrophy with very strong hippocampal loss and medial temporal lobe loss, and loss in other areas, including what we don't see here, the posterior cingulate and other areas. There was a cluster number 4, which was 15% of the patient, the broader patient group, had very focal and quite pronounced atrophy in the hippocampus and other temporal lobe areas, and nothing else. These were 15% of the people. And then 28% of the people were somewhat surprising outcome, or perhaps not so surprising, because this is exactly why we started to do this analysis. They had a very diffuse and kind of uniform pattern of brain atrophy that included the hippocampus and the medial temporal lobe, but included a number of other areas. It wasn't a typical AD-like pattern of atrophy. Now, ADNI, we looked at longitudinal data in ADNI, and we took the late-stage MCI individuals, and we looked at the survival. We formed survival curves. We looked at whether or not they progressed from MCI to AD, whether they became demented, and over a long period, some of them reached out to 8 or 9 years. And what we found is that this group here, with a normal structure, had the best outcome, the red curve. These individuals here with a typical AD pattern had the worst survival curve, the green, all expected. Now these individuals here with the focal atrophy and everything else being normal, initially had a pretty good survival curve, the same one as these subjects over here. But then at some point they rapidly deviated, they went down, and they reached exactly the same level as these individuals over here. So now we can make a hypothesis that these are people who have early stage but progressive disease. And that's, of course, very important to know. And then these people had perhaps the most unusual survival curve, this blue one. Initially, they had a steep decline, about half of them, and then most of them remained stable for years. So again, this atypical pattern may be associated with other pathologies or with mixed pathologies, which is most likely. But definitely these people are different. And again, this all has implications about enrolling into clinical trials, about prognosis and so forth. Now, we applied the same methods more recently to the Phenome Consortium. We restricted the analysis to people who are less than 45 years old to avoid the chronicity effects or the aging effects the accelerated aging effects in chronic patients. This is work led by Ganesh Chand and Greyers in my group and Dom with the Munich group. And we found, we basically found that there were two very reproducible I want to say clusters, but again, using the Hydra method, this is essentially two very reproducible patterns in which schizophrenia patients differ from controls. And the adjusted RAND index, depending on which experiment you're looking at, was in the range of 0.65 to 0.7, which is a fairly good ARI. We did what Andrea suggested. We did random permutations, and we formed a null distribution, and these two clusters were highly significant. For other clustering efforts like k equals 3, trying to find 3 clusters, we find non-significant and less reproducible clusters. And interestingly, for a larger number of clusters, we found highly significant clusters, but not so reproducible. We also noticed that these tend to line up with sex, age, size, so we didn't like that, so we don't believe that. We need to look at it more carefully. But for k equals 2, we find a very, very robust, reproducible effect that was robust to cross-validation, to leave one side out, cross-validation, adjustment for medications, split sample experiments, permutation tests, and looking separately at men and women. So we truly believe this. The data tries to tell us that not all the patients are the same, which we expected, but not to this degree. Let's see what these clusters look like. 2/3 of the patients clustered into this one, which had diffuse and significant volumetric reductions in a number of areas that we expected, including temporal perisylvian regions, orbitofrontal, medial prefrontal, and so forth. So this is what we expected, but it was only 2/3 of the people. 1/3, roughly 1/3 of the patients had no abnormalities, completely normal brains, except they had increased striatum and increased internal capsule. The first subtype, if you look at them against disease duration, the gray matter, the cortical gray matter, was going down with disease duration. The other group remained stable with disease duration. So potentially this might have a more progressive neurodegenerative disease. What is interesting, what's puzzling, is that these two groups didn't differ in anything other than marginally education, where subtype 2 had more education. They didn't differ in positive or negative symptoms, in medication, in disease duration, in sex composition, in age, and so forth, which to many of us was disappointing, but I actually see it as more interesting than less, because it tells us that what we cannot, there is something in the data, in the biology, in the anatomy in this case, that we don't see in the clinical manifestation of the disease. And this makes it very interesting. We want to understand what it is. But the one thing I would like to point out is go back to the effect sizes between patients and controls that we published, and Enigma and other studies have published. We failed to recognize that some of these findings, the positive findings that go in completely the opposite direction, come from 1/3 of the people, and these come from 2/3 of the people. It's not that all of these come from all of the patients, so there's not one stereotypical imaging signature of schizophrenia. And that's very important to have in mind for many reasons, including, of course, enrollment into clinical trials, patient prognosis, and precision diagnostics more generally. So Finally, the last method we have been using, and I don't have much time to talk about this one. This is a method using orthogonally projective and non-negative matrix factorization, which was developed or adapted rather by Aristoteles when he was in my lab. He's now faculty at WashU. And it's a method that has, in a couple of prior publications, the original one in Neuroimage and the PNAS following with the study of brain development along with Ted Satterthwaite. We found very robust and reproducible patterns of structural covariance using this method. In a normal population, you need about 20 of these patterns to represent most of the interesting variants. If you add patients with schizophrenia, you have a lot of more variability, so you have about 34 or so patterns of covariance. And this method is great because not only it clusters voxels into patterns, but it also allows you to cluster patients if you look at the coefficients. I don't have time to go through this. I encourage you to go through Aris's poster if you can make it through that very crowded area. But what I like about these patterns is that they have solved our problem of multiple comparisons. We can now do Bonferroni correction. We don't have to worry about FDR or another multiple comparison correction. We do FDR, and we get p-values of 10 to the minus 6 and 10 to the minus 5. So we're super happy about that because we don't have to defend the multiple comparisons. These are a number of the structural covariance networks that are most significantly affected in schizophrenia. And again, I don't have the time to go through heterogeneity analysis here, but you can talk to Aris or me afterwards. So in summary, and again, we view in Phenom and in other consortia, we have another consortium in aging, we call I-staging, with a much larger number of MRIs. But the idea is not to cluster patients into subtypes, but more to develop a dimensional neuroimaging system where perhaps if you are more along this axis, you have more of this pattern, or you express more of this pattern if you're along this axis, and then maybe you have some of the structural covariance networks involved, or more or less. But this way we can reduce a very complex set of data, and again we've applied this to functional connectivity data, to amyloid images, tau images, is the same method. You can reduce very complex data down to a small number of axes of dimensions, but very informative. You can now deal with maybe 15 or 20 dimensions and apply and look at your data in great detail. And all of this goes after precision diagnostics, patient certification for clinical trials. If there are two subtypes, you would like to look at these people separately from these people for different treatments, because it would be a disaster to lump all of this in the same bucket and try to detect treatment effects, prediction of clinical outcome, and so forth. So I would like to thank a number of people in my group and a number of collaborators in the Phenom Consortium, as I said, and we're always keen on enrolling more members to the consortium. Thank you very much.